Coevolution is reciprocally inflicted evolutionary change between interacting species driven by natural selection, Futuma and Keyes 1992, Thompson 2005. However, there is a continuum of coevolutionary interactions that vary in their intensity from tightly coevolved mutualisms between microbial symbionts and their hosts to generally weaker interactions between free-living organisms such as plants and herbivores, flowers and pollinators, and parasites and hosts. See Chapter 6. The term coevolution was first used to describe gene-for-gene interactions between plants and plant pathogens, Mode 1958, 1961. However, coevolution was popularized when Ehrlich and Rabin, 1964, published their essay on interactions between butterflies and their host plants. They imagined a scenario whereby plants and herbivores exert reciprocal selective pressure on one another, resulting in evolutionary change in both participants. As plants erected new defenses, herbivores ultimately countered with offensive breakthroughs and the arms race was on. Key in their coevolutionary scenario is that novel defensive breakthroughs in plants and offensive innovations by herbivores create temporary, adaptive zones, where plants free of herbivore attack and herbivores with novel offenses are favored, which then promotes speciation and diversification. Subsequently, the Ehrlich and Rabin model has been hailed as escape and radiate coevolution, Thompson 1989, 2005, Futuma and Keyes 1992. Thus, Ehrlich and Rabin interpreted coevolution in a far broader sense than did Mode 1958, who emphasized a gene-for-gene correspondence between the defensive traits of plants and the resistance-breaking adaptations of pathogens. Much of what we have discussed so far has taken a broad view, emphasizing the interplay between plants and herbivores, how they have shaped each other's evolution, and the various defensive strategies of plants and counter-tactics of herbivores. However, regarding all the contingencies that single plant and herbivore species face, how many specific pairwise species interactions are indeed strong enough to exert reciprocal selective effects strong enough to promote speciation and diversification? If, for example, a herbivore is rare and polyphagous, how strong can interactions be between a consumer and one of its many host plant species? Moreover, if a plant must contend with a barrage of pathogens, physical stresses and allocations to growth and reproduction, as well as defense, what is the relative strength of selection from a single herbivore species? One can imagine instances in which plants and herbivores have little reciprocal impact. Alternatively, coevolution is most likely to occur when a herbivore is abundant and monophagous and its host plant harbors few other herbivores or pathogens, Futuma 1983. Therefore, a continuum of possibilities exists that range from tight cobalt associations to loosely linked interactions, Thompson 2005. Also, authors often use the term coevolution without establishing that evolutionary change has resulted from reciprocal selection. With this in mind, it is a valuable exercise to read original papers and examine the legitimacy of the coevolutionary claim. Often, suggestions are made that coevolution has occurred without any hard evidence, and it is particularly difficult to establish that plant traits have evolved in response to selection by insects. Thus, before examining the evidence supporting coevolution and exploring its consequences for speciation and diversification, we need to better understand the variety of interactions that are subsumed under the guise of coevolution. There are many studies demonstrating the one-way adaptation of herbivores to plants or vice versa, Fritz and Sims 1992, Schonely and Bergelson 2000, Berlocker and Feder 2002. The evolution of resistance-breaking genotypes of crop pests that allow them to feed on formerly resistant varieties is an example, as are the many cases of host race formation. However, without evidence for reciprocal effects, the adaptation of a herbivore to a changing plant resource is not coevolution and simply represents resource tracking. As an example, the soapberry bug, Jadera hematolema hemiptera, Rhopalidae feeds on the seeds of plants in the Sapindaceae and has rapidly evolved shorter piercing sucking mouthparts in response to the introduction of golden rain tree, Colrutaria elegans, in parts of the southern United States. Carroll and Boyd 1992, Carroll et al. 1998. The native host plant of soapberry bugs is balloon vine, Cardiospermum corundum, which has relatively large fruits. On this native host, the distance from the outside of the fruit tub centrally positioned seeds is large and bugs have long beaks that can pierce the fruit and access the seeds. In contrast, golden rain tree has small flat fruits and in areas where this invasive tree has established, bugs have evolved shorter mouthparts, while the other dimensions of body size have remained unchanged. Beak length in the two races of bugs is under genetic control, thus providing the raw material for selection and local adaptation. In this system, however, there is no evidence yet that soapberry bugs have influenced plant traits. Most models that portray reciprocal impacts between plants and herbivores, as well as hosts and parasites, describe pairwise coevolution or coadaptation between two interacting species, Thompson 1989, 2005, Futuma and Keyes 1992. The analogy of the coevolutionary arms race often applies to this model, as do examples of gene-for-gene interactions. Perhaps one of the best species-level examples of pairwise coevolution involves an array of leaf beetle species in the genus Blepharidia chrysomelody that feed on a diversity of trees in the Burseraceae. Becerra 2003. By calibrating the timescale of the molecular phylogenies of Bursera and Blepharidia beetles, it was shown that examples of defense and counterdefense have synchronous times of origin. Older lineages of tree species employ a system of pressurized canal-borne resins in their leaves that when damaged produce a squirt of resin that mires non-adapted herbivores. Older lineages of beetles exploiting these trees have countered by cutting the leaf veins, which deactivates the defense. More recent lineages of trees don't squirt, but have evolved a different defensive strategy consisting of complex mixtures of terpenes. Younger lineages of beetle species don't cut veins, but are able to metabolize the complex mixtures of defensive chemicals. Because Blepharidia beetles are monophagous and among the most abundant herbivores in the system, this scenario is consistent with the view that tree and beetle traits have co-evolved in pairwise fashion in response to concurrent reciprocal selective pressures. Most species, however, do not exist in nature as isolated pairs, but as multi-species assemblages. Thus, diffuse coevolution refers to the situation whereby a suite of species exert selection on another species or suite of species, resulting in reciprocal changes that cannot be attributed to a single pairwise interaction. Futuma and Keyes 1992, Inoue and Stinchcomb 2001, Stinchcomb and Roscher 2001, Thompson 2005. For instance, a multitude of herbivores and pathogens may select for high tannin concentrations in trees that in turn foster a variety of different counteradaptations in the community of herbivores. Diffuse coevolution proceeds if the effect of multiple herbivores is non-additive such that plants respond to the net selection imposed by the consumer assemblage at large. A variety of ecological mechanisms can generate non-additive effects that enhance or diminish the effect of any one herbivore on the plant. Younger and Bergelson 1998. For example, a widespread non-additive effect results from induced resistance when prior attack by one herbivore or pathogen diminishes attack by another. Carbon and Baldwin 1997, Viswanathan et al. 2005, Stute et al. 2006. Clearly, the intensity and frequency of a specific plant-herbivore interaction, and thus the opportunity for coevolution, can be affected by the presence of other players in the system. Reciprocal selection on a specific pairwise interaction often varies among populations because the presence of other species changes the strength of the interacting pair. Thus, pairwise interactions may be subject to reciprocal selection only in certain local communities. These coevolutionary hotspots are embedded in a matrix of coevolutionary cold spots resulting in a geographic mosaic of coevolution, according to Thompson 2005. A purported example involves the woodland star, Lithophragma parviflorum, saxifragaceae, and its specialist herbivore pollinator Greya polytella in the Rocky Mountain region of the United States, Lepidoptera, Prodoxidae, Pelmer and Thompson 1996, Thompson and Cunningham 2002, Thompson 2005. With their specialized long ovipositors, moths deposit their eggs in the flower's ovary through the deep corolla tube and in so doing passively pollinate the flowers. Larvae feed on a small fraction of the developing seeds within each inflorescence. Thus, moths impose a cost to plants for the pollination benefits they provide. In some populations, flowers are also visited by copollinators like bombolid flies, Diptera, Bombylidae, which in some cases are as effective as moths at pollination. Copollinators don't oviposit on plants and therefore impose no cost on the plant. Where copollinators are abundant, pollination benefits provided by G. polytella are outweighed by the cost of seed consumption. However, where copollinators are rare, there is a net benefit to moth pollination. Thus, there is a geographic mosaic of coevolution between moths and woodland stars mediated by the presence of copollinators. The interaction changes across the landscape from one of mutualism, coevolutionary hotspots, where copollinators are absent, to one of antagonism where copollinators abound, coevolutionary cold spots. More accurately, this example might be called a geographic mosaic of selection potentials because woodland stars have apparently selected for long ovipositors in moths, but the reciprocal effects of moths on lithophragma evolution have not yet been shown. Escape and radiate coevolution is a special case of coevolution, whereby an evolutionary innovation by either partner creates an adaptive zone that promotes speciation and subsequent diversification of the lineage. Ehrlich and Rabin 1964, Futuma and Keyes 1992, Thompson 2005. An example would be the evolution of a novel defensive trait that liberates a plant lineage from its herbivores, resulting in a radiation of species in that plant lineage. Support for this hypothesis would come from starbursts of speciation, associated with the evolution of novel defenses or counteradaptations in clades of herbivores and plants. Thompson 2005. So, what evidence do we have that rapid diversification in either plants or their associated herbivore lineages follows an adaptive innovation? The independent evolution of latex and resin canals has occurred in 16 lineages of plants, including milkweeds, figs and composites. Farrell et al. 1991. Moreover, evidence abounds for the effectiveness of canal-bearing plants in deterring many insect herbivores. A comparison of sister lineages of canal-bearing and canal-free plants shows a significant pattern, namely that canal-bearing lineages are far more diverse in species, a pattern which is consistent with escape and radiate coevolution. If selection resulting from a pairwise interaction promotes sufficient genetic change in both partners, cospeciation can result. Futuma and Keyes 1992, Thompson 2005. Cospeciation has been invoked in tightly covolved mutualisms such as those involving insect herbivores and their symbionts, plants and their protectionist ants, and obligate plant pollinator systems. Clark et al. 2000, Itino et al. 2001, Kawakata et al. 2004, Hosokawa et al. 2006. However, cospeciation could be causal, in which case genetic divergence and speciation in both parties is promoted by reciprocal selective pressures, coevolution, or it may be coincidental, in which case one partner undergoes genetic divergence as a consequence of selective forces external to the interaction, and speciation in the other partner simply follows. Historically, cospeciation between pairs of plants and herbivores has been extended to entire lineages to suggest that phylogenetic relationships within a diversifying group of insect herbivores might mirror those among their radiating host plants. Futuma and Keyes 1992, Mitter et al. 1991. Thus, in cases of strong reciprocal selection and diversification, plant and herbivore phylogenies might match with ancestral insect species exploiting ancestral plants and derived insects using derived plant species. Parallel cladogenesis then represents instances where corresponding lineages of interacting species groups simultaneously diversify. Parallel cladogenesis was once viewed as the ultimate support for coevolution. Early in the development of coevolutionary theory, Ehrlich and Rabin's model of escape and radiate coevolution was often overextended to mean cospeciation or parallel cladogenesis. Since its early inception, and with the development of molecular tools for phylogenetic assessments, there have been many studies seeking evidence in support of parallel cladogenesis, see Winkler and Mitter 2007. Like cospeciation, however, parallel cladogenesis could be either causal or coincidental. Several criteria are necessary to support a parallel cladogenesis hypothesis. First, there must be significant cladogram phylogenetic tree concordance between the corresponding lineages of players, and second, evidence is required that lineages diverged contemporaneously, Mitter et al. 1991. So, let's examine the evidence for such criteria, explore which groups of organisms show parallel cladogenesis or not, examine factors that preclude codivergence, and comment on the mechanistic link between coevolutionary forces as drivers for parallel diversification. The best evidence for cospeciation and parallel cladogenesis comes from mutualisms involving hosts and their symbiotic microorganisms, Moran et al. 1993, 2005, Clark et al. 2000, Hosokawa et al. 2006. In many cases there is almost perfect cladogram concordance and molecular clock data that support contemporaneous divergence. For instance, molecular phylogenies Ophiorlucan aphid species and their endosymbiotic bacteria Buchnera show very significant phylogenetic incongruence, Clark et al. 2000, figure 4.18. The reason underlying the robust pattern of cospeciation in Buchnera aphid associations is that phendosymbionts are vertically transmitted from mother to progeny before birth and there is no evidence for host switching via the horizontal transmission of bacteria. Also, the bacteria are obligate mutualists and can't live outside their hosts. Moreover, Buchnera benefit their host aphids by supplying them with nutrients, which are required for host reproduction. In fact, Buchnera are genetically specialized to overproduce essential amino acids for their nutrient-limited host aphids, Bowman et al. 1997. Such mutualisms provide perhaps the best evidence that cospeciation and parallel cladogenesis have resulted from reciprocal selection, and thus offer evidence for coevolution. For herbivorous insects and their host plants, phylogenetic concordance is rare and when it does occur, parallel diversification of plant and herbivore taxa is often not contemporaneous but sequential, Percy et al. 2004, Winkler and Mitter 2007. There are, however, a few cases involving Philobrotica leaf beetles Chrysomelody, on mints, see Chapter 6, and Tetraopes longhorn beetles, Cerambicity, on milkweeds that show both cladogram match and contemporaneous diversification, Farrell and Mitter 1990, 1998, Farrell 2001, figure 4.19. For the Tetraopes milkweed association, there is a phylogenetic regression of increasing toxicity of cardinalides in milkweeds from ancestral to derived taxa that is paralleled by beetle diversification, a pattern consistent with a defensive innovation counter-adaptation scenario. In legume-feeding psyllids, Hemiptera, Psyllidae, however, there is remarkable cladogram concordance, but dating of lineages using molecular clocks showed that the plants diversified 8 million years ago whereas the psyllid radiation was backdated to only 3 million years, Percy et al. 2004. Thus, psyllids colonized already existing plant taxa in phylogenetic sequence, giving superficial impression of a coevolutionary scenario. Overall, there are numerous examples of plant-insect associations that exhibit phylogenetic concordance, but fail to meet the criterion of synchronous radiation and coevolution, Winkler and Mitter 2007. Phylogenetic concordance is often precluded because herbivores switch to chemically similar, but distantly related host plants, Miller 1987, Becerra 1997, Becerra and Venable 1999, Kurgot et al. 2005, Winkler and Mitter 2007. As a consequence, patterns of herbivore diversification correspond better to plant chemistry than plant relatedness. Swallowtail butterflies in the genus Papilio, for example, feed ancestrally on plants in the citrus family Rutaceae, but have switched, multiple times in some cases. Relate to more primitive plants in the laurel family Lauraceae, and more derived plants in the carrot Apiaceae, and Astraceae families, Miller 1987, Zakharov et al. 2004. These host shifts in Papilio and other lepidopterans are promoted by phytochemical similarities, coumarins and related compounds, among these plant families, Barenbaum 1983, Barenbaum and Passoa 1999. Although plants and herbivorous insects have certainly influenced each other's evolution, actually demonstrating that reciprocal selection has promoted cospeciation and codiversification has proved more difficult. Evidence for coevolution, cospeciation and parallel cladogenesis comes from tightly linked mutualisms like aphids and their vertically transmitted bacterial symbionts, Clark et al. 2000, and from some parasite and host interactions, Chapter 8. In such associations, the intimacy of the relationship intensifies the opportunity for reciprocal adaptation and cospeciation. Indeed, there are credible examples of pairwise coevolution between herbivores and their host plants, and as expected, the associations involve herbivores that are monophagous on plants that are fed on by few other consumers, Becerra 2003. For most insect herbivores, however, coevolution is often more diffuse and evidence is found at larger taxonomic scales, Thompson 2005, such as that envisioned by Erlachand Raven, 1964, with their escape and radiata hypothesis. Consistent with the escape and radiata model is the fact that elevated diversification in plants is associated with the acquisition of novel defenses like latex and resin canals, Farrell et al. Figure 13. Have had reciprocal influence on each other's evolution. Best, then, is to view coevolution as a continuum with tightly covolved mutualisms at one end of the spectrum and diffuse and spatially dynamic interactions at the other, such as occurs with many plant-herbivore associations, Thompson 2005. Applications. Plant-insect interaction theory in pest management. Between the 1940s and 1960s, changes from traditional agricultural practices in many developing nations led to significant increases in the agricultural production of crops such as wheat, rice and corn, the so-called Green Revolution, Conway 1999. These changes included planting single crops in expansive monocultures, simplifying habitat structure, deploying high-yielding crop varieties and applying copious amounts of fertilizers, all of which predisposed crops for insect and pathogen attack and led to a heavy reliance on pesticides. Benbrook et al. 1996, Norris et al. 2003. For the same reasons, parallel pest problems have occurred throughout North America, Europe, Northern Asia and Australia. As we have seen, planting crops in monocultures and simplifying habitat structure by removing weeds or tilling fields often promotes resource concentration effects and eliminates any advantage of associational resistance. Also, the heavy use of fertilizers promotes rapid growth and high plant quality, which often encourages pest outbreaks and leads to high levels of nitrogen runoff with adverse consequences for neighboring systems. Tillman 1999, Amack and Leather 2002, Boyer et al. 2002. Moreover, due to trade-offs between growth and defense in plants, high-yielding crop varieties were often left defenseless and became very subject to pest attack. Fritz and Sims 1992. For example, there is an inverse relationship between yield and the alkaloid content among the different varieties of tobacco. Vandenberg and Maitzinger 1970. To keep insect pests at bay, pesticides were applied heavily and frequently, which often led to the rapid development of pesticide resistance and the need for more effective pesticides. Thus, toxicologists were placed in the precarious position of developing new pesticides at a rate faster than pests evolved resistance, and the so-called pesticide treadmill was on a roll. Benbrook et al. 1996. The combination of pest resistance to pesticides, the kill of natural enemies, frequent pest outbreaks, rising production and management costs, increasing risks of pesticide contamination to human health and the environment, and public outcry has led to the development of more ecologically based pest management practices and ultimately to pesticide reductions and more sustainable agriculture. Hoffman and Carroll 1995, Norris et al. 2003, Gur et al. 2004. Currently, Integrated Pest Management (IPM) employs an array of complementary control measures to suppress pest herbivores in agricultural, forest and urban systems. Norris et al. 2003, Gur et al. 2004. Recently, more emphasis is being placed in agriculture on ecological engineering, namely the design of cropping systems for pollution avoidance and resource conservation. In this context, an understanding of plant-herbivore interactions has led to more ecologically sound pest management practices in several ways, including deploying resistant varieties, managing vegetation structure by enhancing associational resistance and managing habitat structure to encourage natural enemies, all of which can reduce dependence on pesticides. Strategies for encouraging natural enemies will be discussed in far greater detail in Chapter 7 on predator-prey interactions. Specifically, a greater understanding of the molecular pathways involved in the synthesis and expression of constitutive and induced defenses has led to the genetic engineering of varieties and transgenic crops resistant to insect and pathogen attack. Gould 1998, Cerda and Pauletti 2004, Christou et al. 2006, Ferry et al. 2006. Moreover, techniques are being refined that allow for the expression of resistance traits in the specific tissue or crop stage fed on by the target pest, thus minimizing the migration of transgenes to other plants. Admittedly, concern remains over the use of transgenic crops, but this management technique is in its infancy and much of its potential as a safe control option is yet to be developed. Dively 2005, Ando and Zweilen 2006, Christou et al. 2006. Likewise, advancements in our understanding of the signaling pathways in plants have highlighted the possibility for using natural elicitors such as jasmonic acid as management tools to induce resistance and suppress pests in agricultural crops. Toller 1999A, B, 2002A, Kessler and Baldwin 2002. Moreover, developments on the genetic basis of resistance breaking mechanisms in herbivores are providing insights into how, and how fast, pests adapt to resistant varieties and to improved resistance management strategies. Hare 1994, Gould 1998, Norris et al. 2003. Ecological principles stemming from plant-herbivore interactions have also played a significant role in managing the structure of agroecosystems and in designing urban landscapes in ways that enhance associational resistance and minimize pest problems. Gould 1991, Altieri and Nichols 2004, Norris et al. 2003, Gur et al. 2004. Planting and harvest techniques that can improve associational resistance include growing several crops in polyculture, deploying multiple genetic varieties of the same crop, multilinning, strip harvesting such that some of the crop remains and employing no-till practices, whereby some elements of habitat structure persist. Furthermore, habitat and vegetation structure can be managed to encourage natural enemies and increase pest suppression. Tees and Charnke 1999, Landis et al. 2000, Gur et al. 2004, Langelato and Dino 2004. In general, maintaining structural diversity or plant species diversity in managed landscapes often minimizes pest problems because natural enemies accumulate and persist in such habitats. Landis et al. 2000, Gur et al. 2004, Shrewsbury and Raup 2006. This occurs because diverse landscapes often provide refuges for natural enemies during times when fields are fallow or offer resources in the form of nectar, pollen or alternative prey. For example, adding organic matter to irrigated rice fields in Indonesia boosts populations of detritivorous insects, alternative prey, which in turn encourages the retention of generalist predatoricus predaceous bugs and spiders that ultimately suppress populations of pests like the brown planthopper. Niliparbata lugans, Settle et al. 1996. Likewise, the azalea lacebug, Stephanitis pyreoides hemiptera, Tingadi, is far less abundant on azaleas Ericaceae, in complex ornamental plantings consisting of azaleas and many other plant species than itis in simple landscapes comprised of only or mostly azaleas, a pattern that can be attributed to the abundance of spiders in diverse habitats. Shrewsbury and Raup 2006. As a parting thought for this chapter, it is encouraging to know that strong consideration is being given to increased vegetation complexity as a management tool for reducing pest problems in certain agricultural crops, urban landscapes and home gardens. Grisel 2001, Gur et al. 2004, Shrewsbury and Raup 2006. We will return to this theme again and again in this book since managing, disturbing and otherwise fragmenting plant-dominated landscapes has been, and will continue to be, a major endeavor in human history. See chapters 7 and 12. Summary. Insect herbivores occur in many taxonomic groups, and illustrate many kinds of feeding, from specialists to generalists, from chewers to sucking insects and from external feeders to borers. But plants exhibit many barriers to insect attack, offering, for example, a poor balance of nutrients for animals in general, although insects employ various strategies for circumventing nutritional deficiencies, such as feeding compensation, selection of high nitrogen plant parts and mixing sources of food. Plants also develop mechanical barriers to insect consumers with tough tissues, trichomes and waxes, but again insects have adapted to some of these defenses. Chemical barriers in plants are widespread and generally effective against herbivores, and can be divided into qualitative and quantitative defensive compounds, and constitutive and induced defenses. Induced defenses include phytochemicals which can affect neighboring plants, causing allochemical concentrations to rise in neighbors. Again, herbivores have evolved to cope with plant toxins through deactivation, excretion, sequestration and trenching, among others. Plants may also provide the enemies of herbivores with shelter and food, gaining protection by association with the third trophic level, and genetic heterogeneity in plant populations contributes to variation in food sources for herbivores. Both plant stress and plant vigor influence herbivore nutrition, each with benefits to some feeding guilds and deleterious effects on others. Plant defense theory uses optimal defense as a guiding theme, with plants evolving to protect the most costly and vulnerable parts effectively. Conspicuous plants on the landscape are likely to defend against all herbivores with digestibility reducers, but ephemeral and patchy plants have evolved with cheap toxins effective against generalists. Optimizing allocation of nutrients and defenses is also applied to the carbon-nutrient balance of plants and how carbon-based and nitrogen-based defensive chemicals are synthesized according to the nutrients available, just as in allocation to growth rate or defense depending on the nutrient and photosynthetic resources available to plants. The same theme runs in the growth differentiation balance hypothesis on how a plant's strategy evolves to allocate resources to growth, which limits differentiation and defense, and then to differentiation which limits growth. However, plant defense theory is in need of further refinement in order to cover the great variety of plant strategies and constituents. At the level of plant communities, the habitats available to herbivores are highly heterogeneous, involving patchy communities, variable densities, plant species diversity and architectural complexity. Plant resources can be concentrated or dispersed, and associated plants may improve or degrade a local habitat for herbivores. Natural enemies of herbivores are also greatly influenced by plant species composition and density. Conversely, insect herbivores can influence plant distribution and abundance, especially by seed and seedling predation. The evolution of diet breadth, specialization and generalist feeders involves many factors. Of course, females may evolve to optimize the well-being and survival of their progeny, often by showing a preference for certain plants and plant parts favorable to progeny growth, and/or defense. Other factors involving narrowing of host plant use include camouflage against the host background, interspecific competition, and host plant density, apparency and associated plants. Extreme specialization may result in deme formation adapted to individual plants. Broadening of feeding niches may result in small and/or ephemeral plants, a risk-spreading strategy or increasing enemy-reduced space. Host shifting is an important process in the adaptive radiation of insect herbivore lineages, often facilitated by similarities in phytochemistry and habitat use among host plants. Hybridization between plant species may provide a bridge for a herbivore species to access a new host plant. The concept of coevolution invokes the view of an arms race between plant species evolving new defenses, which herbivores later evolved to cope with. The mechanisms are debated, although the concept of a geographic mosaic of coevolution at the population level in which there are coevolutionary hotspots interspersed in a matrix of passive coexistence between host and herbivore appears to capture a realistic perspective on the interaction. Concepts of the interaction of plants and herbivores apply well to landscapes managed by humans, with integrated pest management employing many ecological concepts, and indeed contributing importantly to their development. Question. 5 lateral interactions, competition, amensalism and facilitation. In the preceding chapter, we focused on interactions between phytophagous insects and their host plants and saw how species occupying different trophic levels can influence each other's abundance, distribution and evolution. Other important intertrophic level relationships include predator-prey and host-parasitoid interactions, and these will be dealt with in forthcoming chapters, see chapters 7 and 8. Here we consider lateral interactions, those that occur among individuals feeding at the same trophic level, and how such interactions, e.g., competition, amensalism, facilitation and mutualism, can affect species abundance, distribution and community structure. Because lateral interactions, and in particular competition, have been studied so extensively using herbivorous insects, we begin our consideration of the topic focusing on this group of consumers, deferring our treatment of lateral interactions in other insect consumer groups, e.g., detritivores, scavengers, predators and parasitoids, to a bit later in the chapter. Lateral interactions between insect herbivores can be negative competition and amensalism, neutral or positive facilitation and mutualism. Daman 1993, Dino et al. 1995, Dino and Kaplan 2007, Kaplan and Dino 2007. In competitive interactions, both participants, either conspecifics or heterospecifics, are negatively affected A, whereas in cases of amensalism one of the players suffers from the interaction but the other remains unaffected A, 0. Positive interactions include facilitation when at least one organism benefits from the interaction 0 and mutualistic interactions in which both participants benefit. Bruno et al. 2003, Bortses and Miller 2006. Moreover, mutualisms can involve tightly covalent obligate interactions, such as aphids and their bacterial symbionts, or they can entail much looser facultative relationships, such as generalist pollinators and their nectar source plants. Because of the complexity and often intertrophic nature of mutualisms, e.g., protectionist ansand plants that offer rewards, we devote a whole chapter to this fascinating topic, Chapter 6. There we discuss only positive interactions between organisms feeding at the same trophic level, although the strength of such interactions, and negative ones as well, are often mediated by basal resources, plants, and natural enemies. Deno et al. 1995, Dino and Kaplan 2007, Kaplan and Dino 2007. In the context of traditional community ecology, negative interactions have received far more attention than positive ones, e.g., Lawton and Strong 1981, Schoner 1982, Strong et al. 1984a, b, Dino et al. 1995 and until quite recently have dominated ecological theory, e.g., Stahovich 2001, Bruno et al. 2003, Lil and Marquis 2003, Nakamura et al. 2003. In our forthcoming consideration of lateral effects, we provide evidence for negative and positive interactions among insect consumers and discuss their consequences for individual fitness and population size. Also, we present the traditional view on the importance of lateral forces, e.g., competition and facilitation, and address the mediating role of host plants and natural enemies. Last, we discuss how the historic perspective on lateral interactions is changing in light of recent information and insights, particularly the relative frequency and importance of competition. 5.1 Competition and resource limitation. Most insects have a tremendous potential for population increase and thus the overexploitation of resources. We see evidence for this potential when lepidopterans such as tussock moths, Orgia vetusta, reach outbreak density, which is followed by widespread defoliation, plant death and ultimately the local demise of the moth population. Harrison 1994. Unless a population is limited by other factors, e.g., climate or natural enemies, competition for limited resources, food, shelter and oviposition sites ultimately deters population growth via strong negative density-dependent impacts on birth rate, fecundity, and survival. Thus, competition can be defined as an interaction among individuals, generated by a shared requirement for a limited resource that leads to a reduction in the growth, reproduction or survival of the individuals involved. Began et al. 2006. Several important elements of the process of competition are worth emphasis. First, organisms must overlap in their use of a limiting resource. 2. Phloem feeding aphid species feeding exclusively from the nitrogen-rich terminal leaves on Thesame host plant constitutes resource overlap. If the terminals are in short supply, then the aphids compete for a limiting resource, often with dire fitness consequences. A second consequence of competition is that it is envisioned as a negative-negative interaction with both participants adversely affected. However, there is tremendous variation in the symmetry of competitive interactions between individuals or species ranging the gamut from classic cases in which both parties are similarly affected to instances of tremendous asymmetry where only one party suffers, namely amensalism. Dino et al. 1995, Kaplan and Deno 2007. 5.1.1 Types of competitive interactions. Interactions over a shared resource can involve individuals of the same species, so-called intraspecific competition, or the struggle may entail interactions among individuals of different species, namely interspecific competition. Because conspecifics are more likely to share the exact same resources than members of different species, the traditional view is that intraspecific competition should be stronger than interspecific competition. See Strong et al. 1984b, Dino et al. 1995. We shall explore the evidence for this long-standing tenet of competition theory a bit later in the chapter. Moreover, there are several different forms of competition. Price 1997, Spade et al. 1999, 2008, Began et al. 2006. Scramble competition, also referred to as exploitative competition, occurs when individuals have the same or similar access to a limited resource in a free-for-all results. Nicholson 1954. In such cases, the scramble among individuals for resources often results in resource depletion, with severe consequences for most or all of the competing individuals. A classic experimental example involves the sheep blowfly, Lucilia cuprina, a carrion feeder in Australia. Nicholson 1954. When a few larvae, 30 maggots, were fed 1 gram of carrion, larval competition was weak, larval mortality was low and many adult flies emerged. In contrast, when the larval density was experimentally increased to 80, larval competition intensified. Once 1 gram of carrion, fewer adults were produced and these flies were small with a low reproductive potential. Above a larval density of 200, food was depleted, larvae experienced mass starvation and no adults emerged. Thus, with scramble competition, there is often widespread overexploitation of resources, high-density dependent mortality and reproductive failure, drastic population crashes and an unstable population dynamic, figure 5.1a. In contrast to scramble competition, contest competition or interference competition occurs when some individuals gain access to more than their share of resources and in so doing deprive other individuals of access, either conspecifics or heterospecifics, to requisite resources, Nicholson 1954.